Okay, so chapter 3 is pretty lengthy and it includes a lot of information. And again, normally I would probably spend at least 2 whole class periods going through this information. And again, I realize, unfortunately, in an online environment, it's not ideal to learn. So I appreciate all the effort you guys are putting forth. So what I did is I'm kind of starting about 2/3 the way through the PowerPoint, but I have posted some great videos on the modules under chapter 3. One is on cell structure and function, which takes you through all the organelles in a cell. And that leads into the next video on protein synthesis, which takes you through DNA and RNA creating proteins. So I would encourage you guys to read through your textbook, work through the first kind of half of this PowerPoint, use those videos as guides, and then we'll start right here with DNA synthesis and cell division. So DNA replication enzymes, before a cell can divide, each DNA molecule must replicate itself so that one of each copy can be distributed to the 2 new cells. It involves 2 types of enzymes. Helicases will break down hydrogen bonds between the DNA strands. This will create a fork where nucleotides can be added to both strands. DNA polymerase will attach complementary nucleotides to the exposed strand, and this is called semi-conservative replication, each with 1/2 being old and 1/2 being new DNA. So this is a look at how DNA is replicated. It'll break apart and then we'll have a new strand forming within the old half that was stretched apart. So the cell cycle is divided into interphase, mitosis, and cytokinesis. Interphase is divided into G1, S, and G2, and then mitosis, I call them the PMAT stages: prophase, metaphase, anaphase, and telophase. So if you can remember PMAT, that's the order they go in. And cytokinesis overlaps the last part of mitosis. Cytokinesis is actually the cell dividing into 2 other cells. So here's a look at the cell cycle. You can see that most of the time, a cell will spend most of its life in interphase, which is kind of a growth phase, replicating its DNA to get ready for division, and then a final growth stage before it gets into mitosis. And there's different stages in mitosis. Hopefully you learned some of this in anatomy. I keep saying that, and I hope you guys did. I'm sorry if not. I've taught anatomy at other schools, so I know it's a lot to get through, but you should understand what each phase of mitosis looks like, and we'll go through each of those phases. And then cytokinesis then is the complete splitting into 2 daughter cells. So interphase is the growth phase. Lots of RNA synthesis is occurring. This will take you through those growth phases. The S phase, if a cell will divide, it'll replicate its DNA at this point. And then in G2 phase, the chromosomes will start to condense, and they will consist of 2 strands called sister chromatids joined by a centromere. So this is a look at condensed chromosomes. So this contains DNA coiled within itself, and each half is called a sister chromatid, and they're joined in the middle with a centromere. And DNA will be wrapped tightly around proteins called histones. Cell death is called necrosis, and this is when a cell dies due to deprivation of blood supply. Necrosis is different from apoptosis because apoptosis is programmed cell death, and that's performed by enzymes called caspases. It can be extrinsic or intrinsic, and knocked out mice with their p53 gene removed are used to study cancers and apoptosis. So prophase, metaphase, you can read through here what they will happen in those phases during mitosis. And I'm just going to go through this slide because this takes us through all of the phases of mitosis together. So interphase is growth, and then we get into prophase, where the chromosomes will be seen to consist of sister chromatids. The nuclear membrane will start to disappear, and the nucleolus is no longer visible. So the big thing in prophase is you'll see the nuclear envelope or membrane starting to disappear around these chromosomes that you see in the center. Metaphase is a fun phase to look at under a microscope. It's always really the first one that's easiest to spot. And this is when the sister chromatids are completely lined up along the equatorial plane. So you'll see these chromosomes completely lined up in a straight line. And you'll start to see spindle fibers reaching out to the chromosomes because they'll be getting ready to pull them apart in anaphase. So in anaphase, the centromeres will split and the sister chromatids will separate as each is pulled to opposite poles. And then in telophase, the chromosomes will become longer, thinner, less distinct. The new nuclear membrane will form and the nucleolus will reappear. Division of the cell into two daughter cells is nearly complete, and that will be cytokinesis when it's completed. So again, anaphase, telophase, you can read through what happens during those phases. Mitosis, just another look at anaphase, telophase. The role of the centrosome, this is a structure located near the nucleus of a non-dividing cell. At the center of it are two centrioles, and they will replicate and move away from each other during prophase, and they will be what these spindle fibers will form from to pull apart, to attach to the centromere and pull apart the replicated sister chromatids. So these are centrosomes. It's a pair of two centrioles, and again, they are what will form the spindle fibers and pull the chromosomes apart. Telomeres in cell division, cells can only divide for so long. The loss of the ability to divide may be due to the loss of regions at the end of the DNA called telomeres. And each time DNA is replicated, a little more telomeres lost. Damaged telomeres will activate p53, which induces cell cycle arrest and apoptosis. And cells that can divide indefinitely, like in bone marrow, have an enzyme called telomerase that replicates the telomere. And this is just another. Okay, hypertrophy and hyperplasia. Hyperplasia is growth due to an increase in the number of cells. This is responsible for the growth of most body regions, especially during puberty. And hypertrophy is growth due to an increase in cell size. This is usually what happens when you've been working out at the gym and where your skeletal muscles increase in size. Homologous chromosomes. So humans have 23 pairs of chromosomes, one set from each parent. 22 pairs are what we call autosomes, with one pair being the sex chromosomes. Each pair is called homologous, and they have the same genes on them, but not identical DNA. Meiosis, unlike mitosis, is the process by which two cell division steps will produce gametes, which are the ova, the egg, and the sperm. And this meiosis only occurs in the gonads, the ovaries, and the testes. So these are the homologous chromosomes, the 22 sets, and then this XY, the 23rd, is the sex chromosome. So then we get into meiosis, and we divide it into meiosis 1 and meiosis 2. And in meiosis 1, they have the prophase 1, metaphase 1 crossing over, you'll see happens during prophase 1, where homologous chromosomes will pair up and parts will be crossed over of the chromosome. And you can see that happening here with these two chromosomes crossing over pieces. Then we get into anaphase 1 and telophase 1. And then meiosis 2 proceeds like mitosis with prophase 2 through telophase 2. Sister chromatids will be lined up. And eventually, because we're going through 2 cycles in meiosis, we get 4 cells at the end with 23 chromosomes each because they've been separated out. So meiosis, we start with a cell going through meiosis 1 into 2 daughter cells, and then the 2 daughter cells will separate out during meiosis 2 to get 4 daughter cells. And you can see that these 4 daughter cells will have 23 chromosomes, so they'll have half the normal chromosomes as needed because they're your ova and sperm. So when combined with another ova or sperm, then they'll complete the 46 chromosomes that are needed. So this takes you through the stages of meiosis. Epigenetic inheritance, silenced genes are passed to daughter cells during mitosis and meiosis without a change in the DNA. through post-translational modifications of histone proteins, which again are what the DNA are wrapped around. This is just methylation, so adding a methyl group to cytosine bases. Problems with epigenetic inheritance contribute to cancer, other Fragile X syndrome, and systemic lupus. And this takes us through the end of chapter 3. Again, there's a lot in chapter 3. Let me know if you guys have questions, and we'll see you for chapter 4.